Hello everyone, I'm Pande, a postdoc at University of Turku, Finland, and today I would like to share our challenge that we organized in heart failure predictions using the microbiomes and clinical data in large population of Finnish adults known as FINRISK. Those FINRISK Dream Challenge is the short name of this. We host our challenge in a platform named Dream Challenge. With the Sage Bionetworks, it already hosts over 60 challenges throughout these years, mainly aimed to solve complex biomedical research questions. We launched our challenge in September 2022, and I put the link here and the QR code that you can scan if you want to know more information and details about this challenge. One of the primary questions that we posed in this challenge is about, can microbiome improve the predictions of the heart failures? The underlying motivations of these questions is related to how cardiovascular diseases are a leading cause of death worldwide. And heart failures is one of the common manifestations of this cardiovascular disease. Several studies has indicated that gut microbiota has been shown to impact several mechanisms involved in the development and progressions of the heart failures, including through inflammations, oxidative stress, and endothelial dysfunctions. Several studies also demonstrated that the microbial metabolites, one of the examples is trimethylamine oxide or TMAO, elevated in the patients with the heart failures and related with the increased risk of the cardiovascular disease. Despite of these preliminary results from the cross-sectional studies, large-scale prospective studies on the association between the gut microbiome and the incidence heart failures have been lacking. Here we have access to the national FINRISK cohort data, which has been started since 1972, aims to investigate the risk factors associated with the cardiovascular disease in Finland. Samples or random sample has been taken continuously every 5 years from different areas in Finland. So the individuals are always different for these 5 years sampling. And in 2002, fecal samples was also collected. And ever since, this enabled us to also investigate the role of the gut microbiomes in several incident diseases, including one in our study is the heart failures. Gut microbiome composition were characterized using this DNA extracted from the fecal samples using the shallow metagenomic sequencing. This became one of the unique data set because it has linked to the health registries, enabling us to follow up all individuals for this study, particularly up to 15 years. Over these 15 years, we observed 559 incidence heart failures in the participants. Here, I would like to show you an overview of our challenge. During the challenge, we provided participants with a synthetic dataset as a proxy to our real datasets. And we also provided participants with 3 baseline models for an examples, in addition to the tutorials and discussion forum on technical related on participations and model submissions. We divided our dataset into 3 sets where we use in the 2 phases of the challenge. In the first phase, known as the submission phase, participants can submit up to 5 models, and then the models were evaluated in the real test data. The results were announced in a later part after we received the models. And then on the second phase, known as the test phase, participants can then submit one of their best models, and we evaluated it in the scoring data that we never revealed to the participants. All of the assessment were performed using two matrices known as Harrell C index for model performance and Hosmer-Lamesso for the model calibrations. In addition, we also perform bootstrap 1,000 times on all of the scores that we can use to calculate the bias factors to ensure that the rank were robust. Here you can also see the timeline of this challenge. As I mentioned, we did not provide participants with the real data sets because FINRISK data contains a sensitive personal information from healthcare registers, which cannot be shared without a formal agreement with the THL Biobank, who's currently hosts the data. To generate this synthetic data, we want to preserve the covariance structures while also privatizing the data. The method we use were rank-based inverse normal transformations that I will not discuss further in this presentation. To give you an overview of our synthetic data sets, here you can see that the distributions of the continuous covariates are relatively similar to the real data sets. And even in three sets, it's also have a similar distributions. Same observations can be shown also in these categorical covariates. Here is our final leaderboard. And we can actually see that the top 5 models that we receive actually perform comparably good. However, only 2 of those models have achieved a sufficiently calibrated model, in addition to one of our baseline models. It also confirmed when we performed the bootstrap calculations and bias factor calculations, which shows that only 2 top performing models can outperform our baseline models. This actually underlined that there is a challenge in building a well-calibrated model as well as the metric measurement itself. Here is the overview of the 2 top performing models. You can read more details about the explanation of these 2 models on the wiki page that has been publicly available, and also the code. So both of the model here employing Cox proportional hazard model for risk-score calculations with different approach in the features engineering and selections. Then for FINRISK here employing a modular regularized Cox proportional hazard model where they build a modular features that they select using the elastic net on different seats and later use for the Cox proportional hazard models for the risk predictions. SB2 team here use the lasso regularized Cox models to select the most important microbiome features. They use different approaches for the feature engineering, including the calculations of the co-abundance. They also define unpenalized clinical covariates such as age, BMI, and others. And after these feature selections, they use the selected features for the Cox proportional hazard model for the final risk predictions. In the post-challenge phase, we work closely with these two top-performing teams to further refine their models. We also perform a model ensembles using the simple mean aggregations. And then from here, we can actually see that the top 2 and 3 combinations perform well, both in terms of model performance and the calibrations, and it slightly improved the single model's performance. We also further investigate the most predictive features to discuss the clinical relevancy of these models. From our observations, we can see that the clinical covariates are stronger predictor for the heart failures, and microbiome features could actually offer a supplementary predictive value to improve the model performance. Specifically for the microbiome's related features, we observe several importance of the species that has been known as the inflammation signature, also TMAO signatures. We also observe a negative association between the alpha diversity index with the incidence heart failures. From this challenge, we actually learned that this challenge of an avenue to explore the potentials of the microbiome-based biomarker to complement clinical risk factors in predicting individuals with an elevated risk of the heart failures. The crowd-search challenge that we organized actually provides a unique platform for collaboratively solving scientific problems. This also highlights the need for further investigations into model ensemble approach to enhance the overall predictive capability and clinical relevance of the models. The synthetic dataset has proven beneficial for challenge participants and could become an option for data sharing while ensuring the privacy protections. I would like to close this presentation by thanking all of the collaborators involved in this project. This project was initiated by the Machine Learning for Microbiome's Cost Action Working Group 2, led by Leo Lahti. And we also like to thank all of our funders, Informatics for Lab and COS, to make this challenge happen. Importantly, I would like to thank all the participants of the FINRISK 2002 and also FINRISK Dream Challenge participants, especially SB2 and Denver FINRISK Hacky Team, who has been active in this post-challenge phase. With that, I would be very happy to have some comments and questions, and we can discuss further during the conference. Thank you.